In Lecture 5, we're going to talk about the biophysics of gas transport. And so we just finished the biophysics of fluid flow. Now we want to move on to another type of flow, and that would be airflow. So in the human body, the best example of airflow would be through the respiratory system. So a lot of this lecture will be based on just understanding the mathematical foundations for working with gases, like the ideal gas law and related principles, as well as going through the foundations of the physiology of the respiratory system. Once you understand the math and the way the respiratory system works, then we'll be able to combine the two and be able to figure out the diffusion of gases, as well as how lung surfactant works. So just like I said before, I'm going to go through a quick recap first of fluid flow in plants, and then I will go through the ideal gas law, some related principles based on the ideal gas law. And then after we know the different equations that we can use for this chapter, we can move on to understanding how the respiratory system works in our human bodies. So going through respiratory airways from the trachea down to your bronchioles and your alveoli, how we breathe in and out, how gas is transported and exchanged with our bloodstream. And then, of course, once you have all of that background, we'll be able to talk about the biophysics of respiration. And so instead of just looking at how the gas is being exchanged from the capillaries to the alveolar ducts, we'll be able to look at models as to how this diffusion of gases occurs. Then we'll be able to take our knowledge of surface tension from previous chapters and lectures and apply it to lung surfactant to figure out how lung surfactant helps us breathe in and out. So first of all, we'll start with the recap. So all we did in the previous lecture was talk about xylem and phloem, and of course, positive versus negative pressure gradients. As a quick summary of xylem and phloem, xylem is the water conducting channel in plants that transports water and nutrients up, so it's one-way flow only. You have phloem being the channels that transport products of photosynthesis from sources to sinks. So sources and sinks are going to be different depending on what you're looking at. If you're looking at minerals from the soil, your source is the soil, and your sink is going to be your leaves or anywhere else in the plant that requires those nutrients. Of course, that transport is only possible due to pressure gradients. I won't go through the anatomy of xylem and phloem again. That was from the previous lecture, but you should know about that as well. Now, with positive versus negative pressure gradients, we know that pressure gradients cause fluid flow in plants. So for the xylem, it's going to go up, and for the phloem, it's going to go in two different directions. We define positive pressure as a smaller contribution from the roots, primarily driven by osmosis, and it helps balance gravity and is responsible for hydrostatics. So there's usually a steady speed of flow for this. Then you have the main contributor of a pressure gradient, which is called a negative pressure. And so this is caused by the transpiration of water from the leaves to the air. So now you have a lack of water or an increased concentration of solute, which means that it's going to draw water up from the roots or the stalk of the plant so that it can help maintain some sort of equilibrium again. So because of the transpiration of water from the leaves to the air, it causes negative pressure or a transpiration pull, and that is based off of surface tension. So before we talk about the respiratory system, we first have to understand the different equations that we're allowed to use when we talk about gas transport. For example, we'll go through the ideal gas law, Henry's law, vapor pressure, and partial pressure. We also have to know what types of physical principles there are when we talk about gases, just so that we know how to model the respiratory system based on theory. So with the ideal gas law, if you've taken basic chemistry, you should know about this already. The equation is PV equals nRT, and I have this over here on the right in red. Gas can be described by three physical parameters. First of all, gas is going to experience pressure, it's going to be at different temperatures, and it's going to exist in different volumes. So that means your gas needs to be able to be described by those three parameters. So we consider an ideal gas as one that has a low density and has negligible intermolecular interactions. This allows us to model these sorts of gases very easily. And so they're going to abide by the ideal gas law, which is the fact that the pressure times the volume is going to be equal to N times R times T. So let's just take a look at what those mean. The ideal gas law is also called an equation of state for an ideal gas. R is called the universal gas constant. It's 8.31 joules per mole times Kelvin. N is the number of moles, volume is V, pressure is T, and temperature is T. So if you have those, if you have at least 4 of those, you'll be able to figure out the fifth. Now, what is the number of moles N? 1 mole is basically the amount of substance that contains an Avogadro's number's worth of atoms or molecules. So Avogadro's number is 6.022 times 10 to the 23. 1 mole is going to be how much of your gas has that many molecules. Then your molar mass, M, is going to be your atomic mass from the periodic table, which is expressed in grams per mole. So if you want to figure out how many moles are in your ideal gas, you're going to divide little m by big M, in which case you have the total number of atoms or molecules divided by Avogadro's number. Then you'll figure out how many moles of gas you have. So based on the ideal gas law, we're actually able to derive many other different laws from that. How can we do this? By making something constant. So the ideal gas law is PV equals nRT. We can create subsets of equations based on what is constant. If we assume that temperature is constant, we can create another equation. So in this case, we have Boltzmann's constant. It comes from PV equals nRT. We can rewrite little n, the number of moles, as the total number of molecules divided by Avogadro's number times R times T. Then we note that R divided by Na is equal to Boltzmann's constant, which is 1.38 times 10 to the negative 23 joules per Kelvin. So that's one of them. We have another law called Boyle's law. If at constant T, your volume is inversely proportional to P. So in this condition, what we're saying is P times V is always constant. So if P times V is always constant, then what I can assume is that P1, if I'm comparing two different systems, I have P1V1 is equal to P2V2. That's because they're always constant. We then have Charles' law. This is what happens when there's a constant pressure. So if you look at PV equals nRT, if you have constant pressure, what this means is this is a constant. That means N and R are also constants, which means V is directly proportional to T. So if I have V divided by T and push all of my constants to one side, I get V over T equals to NR over P, which means if I'm comparing two systems with each other, I have V1 over T1 equals to V2 over T2. So that is Charles' law. Finally, so I've made temperature constant, I've made pressure constant. The only thing I have left to make constant is my volume. So what happens if V is constant? It means that N and R are also constant, which means that I can have P over T is equal to NR over V, and NR over V is going to be constant. And because of that, I can once again compare two systems. P1 over T1 is equal to P2 over T2 because NR over V will always be the same number. So just know that from the ideal gas law, if you keep something constant, if you keep one of the parameters constant, you should be able to determine another law from that, one of these ones. Now, we've talked about gas and the ideal gas law, but in real life, you don't just have one gas in existence, you have a mixture of gases. And when you introduce a mixture of gases, you have to introduce partial pressure. So regular gas is a mixture of ideal gases. The total number of moles is the sum of moles from all of your different components. So your total N is equal to N1 plus N2 plus N3 and so on. There's a different law that we have to know about. It's Dalton's law of partial pressure. And so that's stated as P equals to the sum total of all of your individual partial pressures. Each component's partial pressure, though, can be defined as follows using this equation in red. So you have Pi is equal to the number of molecules divided by the total number of molecules times the total pressure. So there are a few questions or practice problems at the end of this lecture that deal with partial pressure to give you more practice on how you're able to apply Dalton's law. Now, we do want to take a look at vapor pressure now. So we do know that gases can exist as mixtures, but we also have to know that liquids and gases are two phases of bulk matter, and we should know how they're related to one another, especially now that we finished the fluid flow chapter. So in the case of liquids and gases, there's something called phase transition, and you can transition from a liquid to a gas when your pressure is decreased based on this pressure versus temperature graph, and of course, T is constant, and vice versa. So if I have a liquid and I want to go to a gas, then either I decrease, so from here, I decrease my pressure at the same temperature, or if I start here, I increase my temperature and keep my pressure constant. So those are the two ways that you can you can transition from liquids to gas. There are specific terms for this. If I'm going from liquid to gas, I'm going to be going through evaporation. If I'm going from gas to liquid, it's called condensation. So those are two terms that you should know about. Saturated vapor is going to be vapor that's in equilibrium with its liquid. So in this case, just two points to go over. Saturated vapor is characterized by something called saturated vapor pressure. So it's independent of volume and it rises with temperature. The saturated vapor pressure of water is 17.5 mmHg at 20 degrees and 47.1 mmHg at 37 degrees. So we present these values as known values that you can use for assignments in case you want to know what the saturated vapor is at body temperature, which would be 37. Okay, so moving on, we do talk about saturated vapor pressure inside our bodies as SVP-H2O. So it's a good time to now start looking at what kind of gases and conditions we look at in biosystems. So we usually consider gases under two conditions. One is BTPS, which is body temperature, pressure, and it's saturated with water vapor. So that's why we present the SVT. When we say BTPS, that's physiological conditions at body temperature at a atmospheric pressure of 760 mmHg and full water vapor saturation. Sometimes we might want to look at gases under a different condition, STPD. That means it's standard temperature, which is 0 degrees Celsius, atmospheric pressure 760 mmHg, and you want to consider dry cases where there's 0 water vapor pressure. So in our case, we really do want to focus on BTPS because all of our equations and calculations are based in a living system, which does have a lot of water vapor. So air The air that we breathe and the air that's around us is normally water vapor from evaporation. In respiratory systems, all of your gases are in contact with water and are saturated with vapor at body temperature, 37 degrees. The presence of water vapor is going to change the partial pressures of all other components of gas in your system. So that's when we have to tie in partial pressure. And because of that, we have to consider the partial pressure of water vapor. That's 47 mmHg, which we know from the previous slide. For oxygen, it's 100 mmHg, and for carbon dioxide, it's 40 mmHg inside the human lung. So that's alveolar air. So later on, we do have to consider the differences in pressure between the inside and the outside of your lungs. So gases can dissolve in liquids to form gas-liquid solutions. And why do we need to know this? We want to focus on gases and liquids combined in some cases, because we're leading up to discussions about how oxygen, which is a gas, can integrate into our bloodstream through air exchange. So that would be in your lungs, through your alveoli, through the capillaries. So it is important to understand the relationship between gases and liquids in some depth, so that we can talk about that later on. So of course, gases can dissolve in liquids to form gas-liquid solutions. Your solubility is going to depend on gas, the solvent's pressure, and the temperature of the system. When there's chemical similarity, there's high solubility. Some important terms to know that I won't go through in much detail. You should know the definition of solutions, which is a nice, smooth, homogeneous mixture of two or more substances. You should know what a solvent is, which is a liquid substance that's present in a large amount compared to others. Basically solutes, those are the things inside solvents. You have solubility, which is the ability of the solute to dissolve in the solvent. You have saturated solutions, which means that the solute that's present is at its maximum concentration. It can't take in any more solute. You also have a dilute or weak solution, which means that you barely have any solute and it can take in more. The next thing we want to talk about is molar concentration, C. So from chemistry, if you've taken chemistry before, you'll know that the concentration of a solute or a solvent, you can calculate the molar concentration by knowing the number of moles per unit of volume. And the units are going to be in moles per liter. So we already know that gases and liquids can now mix, but how likely are they going to do so? This is where we end up going through Henry's law. So Henry's law talks about the solubility of a gas, and it states that the solubility of a gas is going to be linearly proportional to its partial pressure above the liquid at low pressures. So what this is telling us is that for example, the higher the partial pressure of your gas, the more it's going to be soluble. So a good way of representing this is to have molar concentration C being equal to some sort of solubility coefficient times P, which is a pressure. At equilibrium, evaporation is going to be equal to condensation per unit of time, because everything is static. So we can consider evaporation to be proportional to the concentration or solubility of a gas in liquid. And do know the difference between the two. Condensation is going to be proportional to density, or the pressure of gas above the liquid. We do talk about the solubility coefficient alpha in that equation in red over here. So C is equal to alpha times P, which is based off of Henry's law. But for different types of gases, especially respiratory gases, there are different values of alpha. So at body temperature and at 1 atmospheric pressure, the solubility of our respiratory gases are as follows. So these at the bottom, nothing you have to memorize. You can simply, either the question will give it to you, or you can use this as a reference in case you want to know what the values of alpha are for different types of gases at at body temperature. So we're working with a lot of gases and a lot of equations. So we should be able to understand what types of units of pressure there are. First of all, it's good to understand that 1 Pascal is equal to 1 Newton per meter second is equal to 1 Joule per meters cubed, which is equal to 1 kilogram per meter second squared, where mmHg is an abbreviation for millimeters of mercury column. So this is just a reference table in case you're looking at pressure and you're given a number with a specific unit. You can use this to convert between different standards. So that's just presented for your knowledge. And the only time it would be useful is if you're working through a problem and we give you a number that is, for example, 5 torr. Then you'll have to convert the torr to whatever is required for you to finish the problem. So let's now talk about the respiratory system. In order to apply all the equations we just looked at to the respiratory system, we need to know what's going on in our bodies in order to see how we can apply the equations and in what ways, or if any modifications are required. So what we're going to go through is the respiratory airways of your respiratory system, what happens during gas transport and air exchange, as well as we're going to go through the physiology of respiration. So in terms of gas transport and biosystems, the best example is breathing and exhaling. So breathing or external respiration in animals transports oxygen from and carbon dioxide into the atmosphere through respiratory canals or airways. Specifically, we're going to try and take in more oxygen to oxygenate our blood, and then we're going to expel carbon dioxide, which is a used waste product from our body. How does this exchange occur? It occurs in the membranes of the lungs, specifically your alveoli. So this is the very tip of your lung. You have your trachea, it branches out into your bronchioles, and then eventually alveolar ducts. So each your alveolar ducts are going to be attached to a whole bunch of alveolar sacs. And those sacs are spherical, or can be modeled as spherical, and provide a lot of surface area for gas exchange to occur. These alveoli are covered in capillaries. So we talked about capillaries last time. Those capillaries cover those alveoli, and you're going to experience air exchange between the two. So in living systems, your gas transport can occur through 2 different mechanisms. There's convective transport, or there's diffusion of gases. So we're going to go through each in the next few slides. Convective transport of gases really has to do with how your gases flow in the air canals of your respiratory system. And is going to be very similar to the flow of fluids through pipes. If we take a look at these 2 diagrams here, air is ventilated due to the pressure difference between the outside atmosphere and the respiratory surface. So what's going to happen is when you're breathing in, your respiratory muscles are going to expand so that you can take in the air, and then they're going to contract when you exhale. The pressure in your alveoli, or your air sacs, are going to fall and then rise. They're going to fall because they're going to expand and flatten out, and they're going to rise because they're going to shrink. During what's called tidal breathing, there are 2 things that can happen. You can respire, or through respiration. The pressure in the alveoli is 3 mmHg below the atmosphere. What this does is it causes a lower pressure inside your alveoli, so that it's going to take in, it's going to expand so that it can take in more air. When it goes through expiration, the pressure in your alveoli is going to be above the atmosphere so that it can expel the air back into the air. So looking at the respiratory airways, air is going to enter your body through your nose or your mouth. It's going to be warmed by your nasal cavity over here or the back of your throat. Once it goes down here, it passes through the pharynx and the larynx. And the vocal cord is going to be two bands of tissue over here that extend across the opening of your larynx. Now after that, you hit the pink part, which is where the trachea starts, and that is called the lower respiratory tract. The upper respiratory tract really has to do with the intake of air, and then once it's inside, we start with the trachea. And so the trachea kind of acts like the aorta, but in the respiratory system, it's the largest vessel. So when it reaches a branch, so that one main branch leads to one lung, it's going to reach your primary bronchi. And then each of those bronchi are going to branch out further into bronchioles. And each of those bronchioles will have a bunch of little sacs called alveolar sacs, which are all connected through an alveolar duct. So please do know the order in which air is going to travel in case there's a question. Now, back with fluid flow, we did talk about resistance to flow. In this case, there also is resistance. So we have to talk about airway resistance. So because air is exchanged between the environment and the alveoli, it's going to flow through a series of tubes. Now the tubes are nasal tract, trachea, bronchi, etc. Your airway resistance is going to be the net resistance of your entire network of passages, just because it's so open. So that's one assumption that we can make. Air flows into your air canals under a pressure difference of delta P. There's a volume of air, delta V, at a specific amount of time. Using Puiseuil's law again, we can calculate Rg, which is the resistance of your airways. So we have delta V over delta T is equal to delta P over Rg. So for a normal adult, Rg is approximately 3.3 centimeters of water per liter per second. Okay, so now we're going to talk about the transport of gases in blood. So we're now moving towards being able to understand how air gets transported from how oxygen and carbon dioxide, for example, get exchanged at the interface between gas and liquid in your body. So respiratory gases like oxygen and carbon dioxide are usually transported from the lungs to tissues through the blood. There are various functions of blood, but before I go through them, I first want to go through a breakdown of what blood is composed of. So probably should have had the green part first, but the components of blood are primarily plasma, leukocytes and platelets, and erythrocytes, or red blood cells. As you can see, plasma and erythrocytes make up the most of your blood, with leukocytes and platelets being a small amount, 1%. Based on that, we know that blood flow from our hemodynamics chapter, erythrocytes and plasma work together to carry blood cells across your body, blood cells and nutrients across your body. So that means it has to play a role in the transport or transportation of oxygen, carbon dioxide, nutrients, and waste products. Next, we take a look at leukocytes and platelets. Leukocytes are white blood cells, and they play a role in immunity, whereas platelets play a role in healing and blood clotting. So those two help with protection mechanisms. Now, the last part is something that comes from transport as well, because you have your blood and you're able to transport hormones and other types of small molecules. What's going to happen is it also helps regulate heat and body temperature. So that's pretty much it for the functions of blood. Just remember what it can do in terms of transport, regulation, and protection. Specifically, we will be talking about red blood cells in a lot more detail now, because red blood cells contain something called hemoglobin. It provides red pigmentation to to your red blood cells, but most importantly, it also binds to oxygen. So our whole body needs oxygen, and the oxygen is going to be provided to us through the cardiovascular system because there will be oxygen in blood. So oxygen is carried in the blood through two different forms. Number one, it can be dissolved in plasma, which is usually like barely anything, like 1%. Then you can have oxygen being bound to hemoglobin. So assuming your capillaries are going to get oxygen and get oxygenated in the lungs, your hemoglobin is going to be bound to a lot of oxygen. Once it carries that oxygen to different parts of the body, it donates or transfers that oxygen to different parts within your body and gets deoxygenated. So we need to know what hemoglobin is. I have a picture over here. Hemoglobin, or HB, first of all, we need to understand that with a red blood cell, it's made up of a respiratory pigment called HB hemoglobin that is capable of binding to oxygen. Hemoglobin here is a tertiary protein. There are 4 polypeptide chains arranged in what's called a globin fold. So there's 2 alpha chains and 2 beta chains. Every single one of these chains has what's called a heme group, which binds 1 oxygen molecule each. So we have this in orange here, 1 heme group, and then it's bound to 1 oxygen molecule. So how does it bind to oxygen? Through the charge on its iron. If you have Fe2+, it's going to bind oxygen, and if you have positive 3 iron, then it's going to release oxygen. So almost all of the oxygen in your blood is going to be transported by hemoglobin. So in terms of what to know, I would understand that red blood cells have hemoglobin, and that hemoglobin at most can carry 4 oxygen molecules each. So specifically, how does oxygen binding happen with hemoglobin? HB can attach a maximum of 4 oxygen molecules, and so if all 4 sites are occupied, we call that full 100% saturation of oxygen. The amount of oxygen is called the oxygen capacity of your blood, depending on how much blood you have. So keep in mind that hemoglobin can be termed oxyhemoglobin or deoxyhemoglobin. So you should be able to know that one is fully oxygenated and the other is deoxygenated. In terms of the binding itself, we should take a look at an oxygen binding curve. So this is a good example of an oxygen binding curve where binding of oxygen to hemoglobin depends on the partial pressure of oxygen in your blood. So for human arterial blood, you actually have a partial pressure of 95 mmHg. This means that your blood is saturated. For venous blood, or blood in your deoxygenated veins, you have 40 mmHg, which is 70% saturated. So taking a look over here, the higher the saturation, which assumedly is over here, there's going to be what's called a reversible binding effect. So just taking a look at the graph here, we notice that there's going to be a high affinity of binding if you have a lot of oxygen. So this is atypical of usual curves. What this means is if you have more saturation, it's going to bind even more. And if you have a small partial pressure, you're going to have a low affinity for oxygen. Now we've talked about oxygen transport in the blood, so that's taking in oxygen from the air and then giving it to blood. Now we have to talk about waste products. So what happens when we're trying to expel or get rid of carbon dioxide from the blood to the air? So carbon dioxide is going to move from your tissues, which process different things throughout your body. It's going to produce CO2, release it in the blood, and then it's going to be carried through your lungs to the air. it's important to understand that 90% of carbon dioxide is transported as bicarbonate ions in plasma through cellular respiration, where this is the equation for it, chemical equation. Molecular carbon dioxide is going to enter the blood and diffuse into red blood cells, where carbon dioxide is hydrated. So just keep in mind that 90% of carbon dioxide will be transported as bicarbonate ions. So that bicarbonate, HCO3 minus, is going to diffuse out of your red blood cells and flow into your plasma. It's going to come out, and then chlorine, or negatively charged chlorine, is going to come into your RBC from your plasma, and we call this a hamburger shift to allow for carbon dioxide to get out. In terms of a diffusion coefficient, we do need to understand that different molecules are going to diffuse at different speeds. And for carbon dioxide, it's a lot easier than oxygen. It's in fact 6 times larger than oxygen. So going through gas exchange in lungs, we can assume that the inhaled air is going to eventually reach and fill our alveoli. So remember, your airway branches are going to branch out and eventually end up with those spherical alveoli. And your alveoli, basically one alveoli is one of these circles, where this is the alveolar duct. And for your alveolus, which is one singular alveoli, a thin membrane is going to separate your capillaries with your exterior gas. And this thin membrane is where all the gas exchange occurs. We're going to talk about diffusion soon, basically how much is going to diffuse and what kind of model can we use for this. And it's going to be a very basic model because there's only so much we can do until you move into upper year courses where you can create better models for this. What we can confirm for the time being is that the greater the difference in partial pressure, the greater the rate of diffusion. So in terms of gas exchange in alveoli, in your alveoli, external respiration occurs. There's going to be gas exchange between your alveolus, which is modeled as a sphere, and then here is your capillary that kind of wraps around that sphere. So gas exchange is going to happen where oxygen is going to be taken in from your exterior air in the alveolus. Note that you have a concentration gradient where the partial pressure of oxygen is greater in the alveoli and less in the blood, because only deoxygenated blood needs oxygen. This means that it's going to create a pressure gradient, allowing oxygen to come in. Then you have your carbon dioxide. There's less carbon dioxide in the exterior air and more in the deoxygenated blood. So therefore, your carbon dioxide is going to go out. And then, of course, after your carbon dioxide goes out and your oxygen comes in, both of those partial pressures should be equalized. An important thing to note is that the carbon dioxide is more soluble than water based on our diffusion coefficient that we talked about before. So it's going to diffuse faster and equilibrate a little bit more faster. Now, what happens when we have gas exchange in tissues? So we're done with the lungs. The blood already has oxygen and it's now carrying that oxygen to your tissues so that it can be used. So this means that the exact opposite effect has to take place. Before, we had a higher pressure of oxygen in the alveoli and less in the deoxygenated blood, so that's why we were able to take in oxygen. In this case, it makes sense that now our oxygenated blood has more oxygen, a higher pressure of oxygen, and our tissues have a lower pressure of oxygen. This means that your oxygen will want to be transferred over through the interstitial fluid into your organs or wherever you want in terms of your tissues. And carbon dioxide is going to be higher in your tissues and lower in your oxygenated blood. And after that, the different pressures are going to equalize, and then you have deoxygenated blood that is going to go back to your alveoli to get filled up again with oxygen. Now, the physiology of respiration gets fairly difficult, especially it can get quite confusing with a graph like this. In terms of understanding this graph, it's meant as a reference. We won't be testing you on it. But usually when you do a respiratory test, you'll have to breathe into a bag or you'll have to breathe into something and you get one of these graphs. So these graphs can tell you different things, especially to your doctor. There are different sections to this graph. First, on the left side, you have lung volumes, and on the right, you have lung capacity. The lung volume is basically how large your lung can get, whereas the lung capacity has to do with how much air you can take in or exhale. The total lung capacity, or TLC, is the maximum volume of your expanded lung. That's usually 5 to 6 liters. You have your vital capacity. Let's see where this is. Oh, so that's TLC from bottom to top. That is the maximum volume of your expanded lung. Not all of it's going to be used, but that's your maximum capacity. Then you have your vital capacity. That's the maximum volume of air a person can expel, which is 4.5 liters. You then have the residual volume, RV, being the difference between your TLC and your VC. You also have tidal volume. Where's tidal volume? Not sure if it's here. The tidal volume, represented as TV, is usually the volume of air that's inspired and expired through tidal breathing, and that's approximately half a liter. You have an FRC, functional residual capacity. That is going to be the volume that your lungs are going to retain after breathing out. So of course, if you're breathing out, your lungs are not going to collapse and become paper thin. There still has to be some residual space in there. And so that is called your dead space. So we have a dead space volume, which is the volume of the airways that you just obviously can't compress. The last thing to talk about is compliance. Compliance is the ability for your lungs and thorax to expand. So what this means is you have a volume increase in your lungs for every unit of intra-alveolar increase. So I explained that a little bit over here, but once again, just be familiar with the terms and understand that if we're breathing out, there has to be some sort of residual volume that cannot be removed from the lungs. So now we have the foundations covered. We know how the respiratory system works. We also know some of the equations that could potentially be used for gas transport and gas diffusion. So we're now going to move into the biophysics of respiration. I'm going to go through the physics of the alveoli. That is our main focus because everything happens there. We want to figure out how gases can diffuse, see what kind of models and equations we can use to represent gas diffusion, and then take a look at lung surfactant and surface tension. So those are primarily the two main physical concepts that we will have to know, diffusion and surface tension. So in terms of the physics of our alveoli, let's first understand what has to be done. In order to exchange gases, about 1 meter squared of lung surface is required per kilogram of body weight. That's a lot of surface. So the interior surface of your lungs is the most extensive surface in your body. In fact, it's about 70 meters squared of space. It contains so our human bodies contain about 300 million alveoli, and that's basically a bunch of little bubbles or sacs. One single alveolus is approximately 75 to 700 micrometers in diameter, so it's important to know that they do vary in size, and your alveolar membrane is about 1 micrometer thick. So the membrane is very, very thin to allow for an easy diffusion of gases. So number one, we're going to focus and we're going to focus our efforts on trying to figure out how gases diffuse through this alveolar membrane. And then we're going to shift our focus to understand how your alveolar sacs can expand and contract while we breathe. So the expansion and contraction of our lungs requires us to be able to stretch that alveolar membrane and compress it. So we need to figure out how that is done and why it doesn't actually destroy our alveoli. And surface tension and lung surfactant are the keys to understanding its elasticity. Okay, so looking at the alveoli, it seems like there are a bunch of tubes of blood vessels going over tubes of air vessels. So right now the goal is, can we come up with some sort of model to represent gas diffusion between the two systems, the cardiovascular and the respiratory system? In red and blue, we have cylinders of blood vessels, and in pink over here, we have a very thick and large tube that represents your air vessel. So remember how we had modeled each tube separately? We had modeled the capillaries separately as one cylindrical tube and applied Poiseuille's law. We now have another tube. So it means we could potentially use two cylinders on top of each other or inside each other to model air exchange. And this leads us to something called Krog's model. So we don't have to do any calculations with Krog's model, but it is important to understand what it is. Krog's model makes a very simple assumption. It's a very generalized assumption that your tissue is a cylinder of length L and radius R2. Then you have a blood vessel of radius R1 that passes along the axis of the tissue or cylinder. So keep in mind, this is a model that's best used for gas exchange in tissues, but can also be expanded towards gas exchange in your lungs. So it's a very big generalization, but essentially it's a cylinder inside a cylinder. And so the diffusion of gases is going to take place at the interface of the vessel and your tissue. Now, taking a look at the diffusion of gases in your alveoli, we have talked about Fick's law of diffusion already. We're going to focus on that again. Gases are going to diffuse through the membrane between your pulmonary blood and your alveoli, and between your blood and tissue cells. So there's two different areas where gases can diffuse. Diffusion has always been a kinetic process. Basically, your molecules are moving from an area of higher concentration, like this, to an area of lower concentration. Now, the distance that it travels is delta x, and the flux, or the speed of your flow per unit volume, is going to be your flux, J. So we call it diffusion flux, and we define it as the amount of substance diffusing per unit of time, per unit of area, normal to the direction of diffusion. So in this case, how many molecules are passing through a specific cross-sectional area of a tube at a specific amount of time. So one thing we can assume is that your flux will be proportional to the concentration gradient, delta C over delta X, and you must have some sort of diffusion coefficient, D. So we can simply say the flux is equal to some sort of D times the concentration gradient. So we've modified Fick's law slightly so that we can say this is Fick's first law of diffusion, which is valid where the diffusion coefficient is constant. So we have J is equal to minus D delta C over delta X. Now, we know Fick's law from before. We also know that at the beginning of this lecture, we talked about Henry's law. According to Henry's law, the concentration of gas solute in solution is going to be proportional to its partial pressure in a gas phase above the liquid in equilibrium. Simply put, your concentration of gas is equal to some sort of constant, alpha, multiplied by P. And alpha is your solubility coefficient. So if you combine Fick's law and Henry's law together, you get a combined equation where you can say the flux or diffusion flux is equal to minus d, diffusion coefficient, multiplied by the solubility coefficient times delta P over delta X. If you want to simplify it even further, you can assume that K is equal to the diffusion conductivity, which is your diffusion and solubility terms multiplied by each other. Now, we've talked about gas diffusion. Now it's very important that we move on to how your alveoli can expand and contract while breathing. And there's a strong physical basis for this. We're going to look at the interior of the alveoli now instead of the exterior where gas exchange occurs. So the surface of your alveoli has a very thin membrane and it separates air from water in the lungs. Your water molecules are going to be more attracted to each other than in air, and that causes surface tension. And so that surface tension is going to increase as your water molecules come closer together, which is what happens when we exhale and our alveoli become smaller. So I'll explain that in a bit. But primarily, lung surfactant is going to cover this. There's something called lung surfactant that covers the surface of your alveoli and helps to decrease surface tension. So if you think about it, if I have my alveoli and they're like this size and I exhale, they increase. And if I inhale, they increase. If I exhale, they decrease, they contract. But what's to stop it from collapsing completely? In that case, we have to introduce something to help moderate surface tension. And that's done by lung surfactant. So your lung surfactant is a monolayer, basically one layer of lipid molecules. It's a monolayer that covers the entire surface of your alveoli. And what it does is it helps decrease surface tension by allowing us to breathe in and out more easily. Number one, it reduces the effort needed to expand the lungs. Number two, because of that, it's going to reduce the tendency for your alveoli to collapse. And three, it's going to balance the pressure in both your large and small alveoli. So here's just a brief diagram. You have your trachea going to your bronchus, to your bronchiole, to your alveolar duct, which branches out into a bunch of alveolar sacs. One of these sacs is an alveolus. So if I zoom into one sac, it kind of has this structure, and in red, we have what's called lung surfactant. So there's one layer of lipids that completely covers the entire surface of the inside of your alveoli. As an overview of surface tension, we should first know what it is and why we think that the alveoli will collapse without lung surfactant. So surface tension is going to be related to the amount of work required to increase your surface area by a certain dA or delta A. So the work required is equal to gamma times delta A. Typically, it's measured in dynes per centimeter, the force required to break a 1 centimeter film, specifically for biofilms. But in this case, we've just presented some basic values here for your reference. Next, you have cohesive forces between liquid molecules that give rise to surface tension. So these cohesive forces can be seen over here, and we need to understand why it gives rise to surface tension. So molecules at the surface do not have other like molecules on all sides of them. So this molecule inside your solution has interactions 360 degrees in 3D space. So consequently, the molecules up here can really only interact laterally with each other, and they're going to adhere more strongly to those directly associated to them at the very interface, so the air-water interface. Because they are all similar and they only have similar interactions, they're all going to want to connect closely together through cohesive forces and form what's called a surface film. This means that it's going to be more difficult to move an object through the surface than when it's completely submerged. In surface pressure or in surface tension topics, we we do go through Laplace's law quite a lot. So Laplace's law is an equation that helps us calculate the pressure inside the alveolus. So the equation is delta P is equal to 4T over R. What this tells us is that we'll be able to calculate some sort of pressure on the outside and pressure on the inside, and how much tension is required to allow for those two to happen. So T is the surface tension and R is the radius of the alveolus. So when R is very, very small, the pressure difference is going to be very, very large. And what this means is that it can cause collapse. So you have to have some sort of surface tension to allow for stability. So your lung surfactant is the one thing that will provide. It's going to be the one factor that prevents your alveoli from collapsing. And so the alveoli that is not covered with surfactant will collapse when contracting and as it gets smaller, because your tension can't be adjusted. So that means your pressure is going to get larger and larger, and then your alveoli will just collapse. Now, taking a look at surfactant composition, now that we know what surfactant can do for us, which is reduce surface tension, we should probably take a look at what causes this and how is it structured to allow this to happen. So lung surfactant consists mainly of saturated phospholipids. It's just one layer of lipids called a monolayer. Primarily, it's 80% DPPC, or dipalmitoyl phosphatidylcholine. It's a primary component of surfactant. It also contains cholesterol, which helps with membrane fluidity, as well as surfactant proteins. There are actually 4 different types of surfactant proteins. Surfactant proteins A and D play a role in the immune response, so it helps protect your lung surfactant from infection, especially because it's exposed to air all the time. You also have surfactant proteins B and C, which play a role in surfactant spreading, so it helps your surfactant contract and expand. So we call surfactant proteins B and C small hydrophobic proteins. And specifically, those hydrophobic proteins allow your lung surfactant to expand and contract very easily. So how do they work? If you have surfactant proteins B and C in a monolayer for your lung surfactant, the best way to explain it is, if you have one layer of lipids, a monolayer, your lung surfactant, covering your lungs, when it expands, so I'll go through the second one first. When it expands, you truly get one monolayer. But when it contracts, what happens? You have surfactant proteins B and C. It allows for multiple folding or multiple layers of monolayers to be created. So when it's contracting, or when you're breathing out or exhaling, your alveoli are going to shrink, which means your layer of lipids is just going to start overlapping over one another, and these surfactant proteins help stabilize it. So surfactant protein C usually helps stabilize the multi-layer formation, which is what this is called. It helps stabilize multi-layer formation, whereas your surfactant protein B helps with the lateral spreading of your monolayer. So just understand what the different surfactant proteins do and a general description of why your lung surfactant is capable of expanding and contracting. So the main reason for it to be able to reduce surface tension is because it allows for multi-layer formation upon exhalation. After that, there's a bunch of practice problems that you can go through. This first one has to do with calculating what volume the lung expands to due to a pressure difference. The second problem has to do with partial pressure. Same with the third one. So we're trying to find the percentage of carbon dioxide and air inside the lungs. Practice problem number 4 has to do with calculating the volume of expired air within the lungs. So that most likely could be partial pressure and gas diffusion. This one goes through diffusing capacity. It goes through a description of what it expects you to assume. For example, 70% of inhaled air is going to reach your lungs. And of that amount, 5% of oxygen will diffuse across the membrane. So in this case, most likely you'll have to use Fick's law for diffusion to calculate how much oxygen is transported. And last but not least, instead of looking at humans, we're going to extend our knowledge a little bit and take a look at what happens when insects are breathing. So we're going to take a look at insects, taking a look at the oxygen that diffuses from the outside to the tissues through an insect's trachea. Okay, so now going through what we have to remember for this lecture. Number one, you should understand the physiology of respiration. What are the respiratory airways and pathways? What's the alveolar structure? Be able to understand the mathematical foundations of gas transport, so like ideal gas law, Henry's law, Fick's law of diffusion, as well as surface tension measurements. And of course, you should understand the physics of the alveoli. So understand how to apply the equations, understand how to explain what happens with air exchange in the alveoli and in your tissues, and understand how lung surfactant works to reduce surface tension when you're exhaling.